So this begins our second part of the Origin of Species lecture. In the first one, we talked about species and the challenge of trying to define it. Again, for all practical purposes, our definition is going to be organisms that can mate and reproduce fertile young naturally. That means you're the same species. If you can't do that, then we're not going to consider you the same species. So we talked about once species are evolved, there's a bunch of different things that prevent them from coming back together. Those are what we call isolating mechanisms. And the isolating mechanisms that we talked about in the last lecture were these things called prezygotic. They actually block the zygote from forming. So the egg and the sperm never get together. Most of the time, the two individuals never even have the opportunity to mate, so they can't put the egg and the sperm together. But if all those prezygotics are overridden, not in place, or not applicable, there still is the gamete fusion issue, that if you have two different species, the gametes can't actually fuse if the sperm does not contain the correct enzyme or protein structure that enables it to attach and penetrate the egg. So that enzyme on the sperm is built based on the DNA of the father, the male of that species. The receptor protein on the egg of the female is built because of her DNA, and that's what allows her to build that particular protein. So those two species, or those two individuals of the same species, build compatible proteins, the receptor and then the protein on the sperm. But in different species, they're not compatible. Okay, so those are all prezygotic. What we want to explore here are what are called postzygotic isolating mechanisms. So these are going to be isolating mechanisms that prevent the formation or development of fertile offspring. Alright, so the offspring don't develop, they don't form correctly, they are not viable, they cannot pass on their genetics, whatever the case may be. Zygotes have already formed. So our little cartoon here, you have two different frogs, they mate, they produce offspring, then those offspring try to produce further offspring, and you know what? It doesn't work. That individual dies, or those individuals are not fertile, whatever the case may be. So there are several examples of post-zygotic isolating mechanisms we want to explore here. Okay, the first one that we'll explore is what is called hybrid sterility. Okay, so mating can happen. The two individuals get together, the male and the female reproduce, the egg and the sperm combine, but their offspring is not fertile. They're sterile. So you take a horse and a donkey, they can mate, they can produce an offspring called a mule. The mules are primarily sterile. You don't have mules breeding to mules to get more mules. You use horses and donkeys. Horses and donkeys are different species, very closely related, but they can reproduce. Now, naturally, they're probably not going to find each other in the wild, so we are skewing this a bit. But when they reproduce, they have a mule. And that mule is the hybrid, which tends to be sterile. The other image, the lion and the tiger, they produce a tigon or a liger, based on which one's male, which one's female. And I'll show you a really neat chart here in a minute. But you put a lion and a tiger together in a zoo, yeah, they can mate, they can overcome habitat isolation, they can figure out the behavior, the mating seasons match up, the mechanicals fit, and the gametes do fuse. Now in nature it won't happen, but in captivity they do, and they produce ligers or tigons. Those individual cats are sterile. So a gigantic lion with the stripes of a tiger, or a tiger with the lion's mane, those offspring are sterile. It stops there in that first generation. You don't take tigons and make tigons and tigons and produce lots and lots and lots of generations. So hybrid sterility is a post-zygotic type of block that stops two different species from mixing their genetics back together. Okay, our next one here, went too far, is when we have a buffalo and a cow. You produce a beefalo. Okay, so here's the little chart I was talking about. You get a liger, which is a hybrid, from a lion, a male lion, and a female tiger. You get a tigon if you have a female tiger and a male lion. So these are all going to be sterile offspring that we will see develop based upon those two individuals. So mules, horses, and donkeys. If you switch the genders, a male horse and a female donkey, you get a hiney. Beefalo. Some of us may have actually eaten this. Buffalo or a bison and a cow. That's not natural. You don't see cows running around the plains of Kansas naturally, but in captivity we put them together. We create this big, big animal called a beefalo. It is sterile. The Z-donk, zebra and a donkey. Similar animals, different species, but they can come together and reproduce. They produce a sterile offspring. And then you get a whale and a dolphin. Both are mammals. Both can reproduce together, but they produce a wolphin. Naturally, not going to happen. That wolphin is sterile. Okay, so hybrid sterility, again, is one of our examples of a post-zygotic block. The next example is what's called hybrid inviability. Inviable hybrids. The offspring does not survive or develop. Okay, so the hybrid, the individual, here's your egg, your zygote, it starts to divide, keeps dividing, but then it hits a point where the genetic information isn't accurate, it isn't correct, and it stops. The division stops, different stages of cell division, it shuts down, it doesn't continue, and that is no longer viable. So the offspring or the developing embryo won't develop. It maybe develops partially, doesn't survive childbirth, doesn't survive initial phases of its life cycle, whatever the case may be. The challenge is when you're mixing different DNA. So think back to meiosis. And when we talked about chromosomes, you're supposed to have a specific number, a haploid number in the egg, haploid in the sperm, that combines to form the diploid zygote. If the zygote has extra or not enough, it doesn't develop correctly. And the number of chromosomes in every species is what dictates the species genetics. So when you're mixing and you're taking 23 and 24 and getting 27 instead of 26, there's a problem, or 46 I should say. It throws things off during development. It's a natural mechanism to prevent different species from coming back together to try to recreate the ancestral line of the ancestral population. Okay, so now when we're looking at speciation, speciation follows different patterns. So it's generally a two-part process. So what we start out with is you have your population. You have identical populations, and then those identical populations must split or diverge. They can diverge geographically. Maybe they're now on this side of the river versus that side, or this side of the mountain range versus that side. But that's a big thing. The population must split. It's got to diverge, and that's going to create some differences between the environments that the populations exist in. That causes them to experience different reproductive events. Mutations will start to occur. Reproductive isolation has to then develop or evolve to maintain the differences. Okay, so you have to keep these populations separated. So they're on, let's just make it easy, east side and west side of the mountain range. Population got split. The population on the east side is on a dry side. The population on the west side is on a very moist side of the mountain range. If we're talking about plants, now the dry environment, you adapt differently. You evolve differently than if you're on the wet side. But you have to maintain this isolation, causing those populations to stay different and evolve in different directions. If there is this homogenizing effect of gene flow, it can actually erase the differences. So if the genetics are allowed to mix back and forth across the top of that mountain, you're not going to maintain your differences. So those differences need to be maintained for the populations to evolve differently. As they evolve, they start to undergo what we call speciation. Now, speciation is the divergence of a population into different species. This generally happens, or happens easier, in populations that are geographically isolated. So east side, west side of the mountain range scenario. Okay, so let's take a look at, as speciation is occurring, often it goes through what's called allopatric speciation. And this is when there's some type of geographical barrier that splits the population. Okay, so something geographically could be continental drift, it could be this river formed after a major flood and split the beetles on each side, it could be glacial movements, glaciers retreat, glaciers move forward, and this causes populations to get split geographically. If you're geographically isolated and there's some kind of physical barrier, that's what we call allopatric speciation. It helps to maintain or cause these differences to accumulate over periods of time. Now, sometimes there's a behavioral difference. This is known as sympatric speciation. So behavioral differences cause the population to split. All right? So it could simply be sexual selection, female preference. The picture here is showing the Amish. Amish communities maintain a very closed community. They're, for the most part, reproductively isolated. Geographically, no. We can drive to an Amish community today. We could jump in a car and take a trip and go to their community. But the likelihood of mixing our genetics with the Amish community, marrying and having children within the Amish community, is very, very limited, very unlikely. That's a type of sympatric speciation. For some reason, there's behavioral differences that cause a population to split. It could also be genetic differences. And what we'll start to see is certain genes are maintained in certain populations versus other populations. And that causes the populations to change and diverge over periods of time. So as that's occurring, sometimes we see polyploidy happen. Extra genetics show up, extra pieces of DNA. If those extra chromosomes are maintained, that starts to create differences or changes within that particular population. Maybe it's a beneficial change versus a negative change. But sympatric is when you have behavioral differences splitting the population. Okay, there's a couple other types of examples of sympatric. This is more on the genetic variation. So genetic variations occur causing populations to diverge. The first one is called autopolyploidy. In autopolyploidy, all of your chromosomes arise from the single species. So everything you get is from the same species. You're not mixing. It's just within your species. So what we'll talk about in the next lecture is how autopolyploidy and allopolyploidy play a big part in sympatric speciation.